I think it's so impressive, this meeting, because there's a range of people. Many of them obviously are not professional scientists. They seem to be interested and lots of really good questions. And I think an outreach like this is very important, and people should do more of it, I think. A few years ago, I was in one of the small libraries in Cambridge, and I came across a shelf of books that I hadn't encountered before. Most of these books refer to work that was done a century ago, but there was a lot of information in these old studies that I think we should not forget. One of the leading lights was somebody called Herbert Jennings, who wrote in 1918 a book called The Behaviour of Lower Organisms. For example, this is a description of a simple amoeba. Jennings was watching an amoeba that was trying to feed on the spore of another single organism. Jennings observed this for quite a time, 10 minutes or so, and he said it was very difficult not to form the impression that this cell was being frustrated. It was hungry, wanted to get hold of this food, and it was kept being eluded by the spore that was getting out of its grasp. But in fact, the most complicated kind of behaviour was shown not by amoeba, but by ciliates. Jennings described something called a stentor, which is a ciliate, but in fact it lives attached to the surface in the bottom of a pond. It's covered with cilia. At the top end, it has a mouth part, a buccal cavity, and around the rim of this buccal cavity there is a set of specialized cilia which beat continually and sweep water into the buccal cavity. The stentor then decides whether it wants to keep them and digest them or spit them out. And he did this interesting experiment. He came up to a stentor and he brought a pipette that was filled with particles of carmine, which is a dye, and sprinkled them onto the feeding area of the stentor. So the question was, what would it do? Well, it turned out that the first reaction of the stentor was to twist on the stem so that the buccal cavity was pointing away from the noxious stimulus. And it would remain in this position for some time, and then it would come back again. And if the carmine particles were still being supplied, it would try a different strategy. And in this case, all of the cilia would reverse their beating. If this didn't work, then stentor had a third strategy, which was to contract very rapidly. It would contract down to this holdfast and remain there for many minutes. After a while, eventually, of course, it had to feed. It would unravel and grow out again. And if the noxious substance was still being applied, then there was a fourth strategy. The stentor contracted rapidly and vigorously to the extent that it actually broke the holdfast from the base and it floated off into the water. And now the cilia, which had previously been bringing the fluid into the mouth part, was used to swim and eventually come down to the surface, the lower surface of the pond. Now it would crawl around using the specialized cilia at the top as a locomotory organelle. It was trying to search for a new habitat. Eventually it found a spot that it liked. It secreted mucus, formed a new holdfast, and sprouted again. This was a small animal that was doing this, but what you have to remember is that this is a single cell. So the question is, how does it do it? What is it inside this cell that is able to confer on it this ability to behave? This is a cell which has evolved in the same way that cells that form our bodies. So just to summarize, single-celled organisms are aware of their surroundings. They can detect chemicals, mechanical forces, electric fields, visual stimuli in some situations, and gravity. They respond by moving, changing their shape, changing their chemistry, and they do this selectively in a discerning manner in the absence of a nervous system. So how do they do it? And the answer is that they are full of proteins. Proteins are made as chains of amino acids. The amino acids have different chemical properties. They fold up spontaneously, as illustrated here. The protein itself has distinctive properties. So this particular one here is a simple enzyme called hexokinase. It's got a hole or a binding site on its surface, which is just the right shape to bind another molecule, and in this case it's glucose. And the hexokinase is evolved to bind glucose very tightly, and it's also evolved to bind a second molecule, which is not shown here, called ATP. The ATP and the glucose are bound next to each other on the surface of the hexokinase in such a way that a phosphate group from the ATP is transferred to the glucose. And it's not just proximity on the surface alone that does this. The protein also binds in such a way that it stretches particular chemical bonds so that the reaction is very much facilitated. So it's a little transformation machine. And that's a kind of a processing machine, but not very interesting. The interesting part came in 1961 when a group of French scientists presented evidence that proteins could bind more than one component. They were, in their term, allosteric. The reaction catalyzed by a particular enzyme depends not only on the substrate that it's converting, but on something else. It turns out that proteins have on their surface a possibility to have a second site, which can bind a second molecule, and that second molecule regulates the activity of the first. This is a protein called calmodulin that binds to calcium ions, and when it binds to calcium ions, it opens up in the manner shown. When it binds to calcium, it's active. When it doesn't bind, it's inactive. The magic thing about this allostery is that because the protein structure is determined by evolution, it means that any process in the cell, it can be linked and regulated by a second process. Just to illustrate that, at the top we have just a diagrammatic picture of an enzyme, which in its active form will convert A to A prime. In this case, we'll only do it if a second molecule, B, is bound to a different part of the enzyme. Now we've got a second element, which, say we've got a constant supply of glucose. Now you come in with the second regulatory molecule, the B, and depending on whether you raise or lower this, you can raise and lower the production of the product. Now, this is exactly analogous in a formal sense to what a transistor does. This is just a selection of some of the enzymes and other proteins that have been studied. And I've presented them here in the form of a processing unit. It's a format that became used in the early days of artificial intelligence, where people were designing processes to be used in computers. So a simple enzyme certainly has these properties, that it has an input and it has an output. And numerous studies have shown that depending on the conditions can show very complicated relationships between input and output. Now you can put these proteins together in little circuits, and I won't go into a lot of detail, but you can form particularly common is the formation of a switch. Very often in cells you need to turn something on off very sharply, and you can do it by means of the kind of arrangement shown at the top. And of course you can, in principle and in practice, take any of these little circuits and link them together to make more complicated devices. So proteins are large, complicated molecules that bind to and recognize other molecules. They can produce chemical changes, and doing so repeatedly and rapidly. Allosteric proteins, I mentioned, they can link together two or more processes, allowing one process to regulate the other. Protein function is continually modified by chemical modifications such as phosphorylation, and small sets of proteins together can form simple logical operations such as addition, subtraction, coincidence detection, amplification. But those little circuits are themselves put together into whole networks. And to illustrate how this works, I'd like to show you, this is in fact the subject I specialize in working on, and that is bacterial chemotaxis. Bacteria such as Escherichia coli swim by means of flagella, and this flagella are driven by rotary motors which are inserted into the membrane. the reason they swim is to find food. In a normal condition, if there's no food around, they will swim for about a second, and then they will change their direction in a random fashion, and then they move off. Now, this is changed if they encounter some food. If, for example, there's some food over there that's diffusing towards the cell, and it happens by chance to be moving in that direction, then it will stop the tumble and it will keep going in that direction. On the other hand, if it happens to be moving away from the food, it will tumble sooner than it would otherwise. So it doesn't actually ever know which way the food is. By trial and error, it will find its way. But of course, this is a very simple cell, and this is a very simple process. If you look around in living organisms, eukaryotic cells, the cells that of the kind that are in our body, are much larger, much more complicated, and the networks are correspondingly complicated. So cells are full of very complicated networks of biochemical reactions. Now, one feature of them, this sort of network I'd like to just draw attention to, and that is that it has some similarity to what computer scientists have termed neural networks. And the general features of neural networks are actually present in these cell networks, and I'd just like to illustrate that. So imagine that we have, in a cell, we have a membrane, and we have a set of receptors in the membrane. On the left there, we have receptors that are detecting matrix molecules. The receptors on the right are detecting factors called growth factors, which are going to stimulate the cell to grow. Now, let's imagine that each of these receptors is binding to its cognate ligand, and then it passes its signals down to a layer in which there's enzymes, kinases, which are stimulated or inhibited depending on the way that the proteins interact by these receptors. And now these kinases, which then go ahead and add phosphate groups to some other proteins, shall we say a kinase 3 here. Now, what does kinase 3 know about the environment outside the cell? All it knows is what it's told by kinase 1 and kinase 2. And all they know is certain combinations of the receptor activity. So eventually, if you go down far enough, if you go down enough levels, you can see that you can have proteins that are recognizing quite sophisticated features of the environment. Okay, so cells detect features of their environment by receptors in their membranes. Signals from the receptors travel into the cytoplasm and the nucleus, where they are integrated and processed. Protein networks perform tasks such as pattern recognition, feature detection, and the storage of memories. Output signals travel to the locations in the nucleus and the cytoskeleton where they generate cell responses. And so it raises the question of how sophisticated the cell is. And it's interesting to ask, I think, how much they know. So let me address this question by coming back to to E. coli and again to the receptors that I talked about. These are real bacteria which have been stained with a label which has stained the receptors. You can see as green caps to the end. And this is an artist's impression of the receptors themselves. There's usually 3,000 or 4,000 inserted in the membrane. Now, I mentioned that the receptors were multifunctional. And remarkably, these receptors are also used by the bacterium to taste acidity and also temperature, so that the bacterium swimming around will avoid very high temperatures and will avoid very low temperatures. An experiment was carried out in 2007 in which bacteria in a suspension were exposed to a very weak laser spot. If the bacteria were taken from a rapidly growing culture, they would congregate in the warm area, and that's shown by the red cells here. But if the same cells were allowed to grow into a dense culture, and they were put into the same situation, and this is the green cells, they moved out, away from the heated region. Well, the argument is that if you've got plenty of food and you're growing like crazy, then the best thing you can do is to move to a warm place because your division speed will be proportional to temperature. But if you're in a situation in which food is limiting, it's probably better not to divide rapidly. It's probably better to slow down and look for alternative strategies. This illustrates that one of the molecular bases of what's going on, the different kinds of receptors, one heat avoiding, one cold avoiding, shift in their amounts. So I'd just like to finish with this question. Isn't it just like a thermostat in a room? Thermostat typically has a heat sensing element, that's the coil. As this cools, the twisting will bring the connector here. When it contacts the grub screw, it will complete an electrical current, and the electrical current then operates the heater, and the room heats up. As the room heats up, the coil will go the other way, and it will break the contact. Now, this is clearly a device, and nobody would claim that it knows anything. It's just a dumb device. But then you have to think of who made this device. Let's go to the bacterium. Now, this is a diagram which shows a bacterium. Now you have the sensor, the temperature sensor, which is the receptors. And they send their signals, as if it were by a connection, through to the motor, which is the output. The precise temperature range at which this operates and the connections are determined by the details of the protein. If I ask you who designed this, of course, most of you would probably say it wasn't designed, and that's true. But nevertheless, in the course of evolution, exactly the same instructions, if you like, were put into this device as was put into the human-made thermostat. They're all there, but they're there because of evolution. So, features of cell chemistry written in sequences of DNA and proteins represent prior knowledge of the world. Refinements and modifications occur as the cell feeds and grows and moves, and the living cell uses this picture in order to predict future events. So a free living cell has knowledge of its surroundings and uses this knowledge in a thoughtful way when challenged. I'll finish there. Thank you.